Hi, good afternoon everyone. My name is Sheena McCormick and I'm going to present to you today conjugation of phosphanoacetic acid to nucleobase promotes a mechanism-based inhibition. And these are the authors. So this paper is going to discuss how analogs of pyrophosphate such as phosphanoacetic acid and phosphanoformic acid are used to inhibit viral polymerases. So what is phosphanoacetic acid and phosphanoformic acid? Phosphanoacetic acid from here on out will be referred to as PAA, and phosphanoformic acid will be referred to as PFA. Phosphanoacetic acid, PAA, is a small molecule compound. It is a product of DNA polymerization reaction, and it is less effective against viral polymerases, which means that it is non-cytoxic. PFA, on the other hand, is a most effective inhibitor of HIV reverse transcriptase. The sensitivity of the viral polymerases towards PFA is greater than that of mammalian replicative polymerases. However, this is not used very often, only in cases where the HIV and HIV-associated viral infections are critical because it is cytoxic. And these are figures of the PAA and PFA. So what is HIV? HIV is human immunodeficiency virus that it causes AIDS. AIDS acronym stands for Acquired Immunodeficiency Syndrome. This figure right here indicates the AIDS virus. And AIDS is a chronic, potentially life-threatening condition, and it is transmitted through various methods. It's transmitted through sexually transmitted disease, contact of the blood with an infected person, or from a mother to a child during childbirth, pregnancy, or even breastfeeding. In this diagram, you're going to see this blue circle that's attached to this red string. This red string, that is the reverse transcriptase enzyme. And what the reverse transcriptase enzyme does is it transcribes the sequence into a complementary DNA sequence. So here you have your HIV RNA strand, which is single-stranded. The reverse transcriptase comes in and it will read that HIV RNA and make DNA. You then get your single copy of the DNA, a second copy is generated, and then now you have a double-stranded DNA molecule. The reverse transcriptase is also a viral genome that cannot be introduced to the host cell. It is required in order for the virus to reproduce. So what's the focus of the paper? The paper will focus on 3 analogs of the pyrophosphate, and those analogs include these 3, PA5T, PA5TT, and PAT. So the paper states that there is a relaxed specificity of an innate polymerase. It is vital to the survival of the virus. And then thus, because of the specificity, the inhibiting polymerase justifies the essence of the highly active antiretroviral therapy, HAART. This is a revolutionary approach ensuring control over HIV. So in order to focus in on how these inhibit the polymerase, they used one form of method called native PAGE assay, PAGE standing for polyacrylamide gel electrophoresis. This is similar to the SDS that we used during the molecular lab. What happens is chlorine ions and glycine ions are used as a front. This will form a boundary that stack and separate polypeptides by charge to mass ratio. And so here you'll see a micropipette being used to insert the sample into the stacking gel. And then this top part is the stacking gel, bottom section is the resolving gel. It is then ran until the dye reaches the bottom. And then once the dye reaches the bottom, the gel is removed and stained so you can then visualize your proteins. So using that process, proteins are prepared in a non-reducing, non-denaturing sample buffer. This maintains the protein's secondary structure and the native charge density. Multiple bands are visible if the target protein has polymerization forms in the sample. And most proteins are acidic and slightly basic, so they will migrate towards the negative polar. However, if the protein is highly basic, then one will need to reverse the anode and then run the gel. So a question that was raised in this question, or in this paper, was, does binding of the polymerase to DNA substrate change conformation of the ternary complex? So what they did was they used two natural nucleotides, NTs, and those two natural nucleotides are the DTTP and the DATP. They also used two analogs, nucleoside triphosphates, which are the PA5T and the PAT. So enabling detection of both specific and non-specific complexes will help answer this question. So what they did is, by doing what I mentioned before and using the native page assay, they then found the results of, in the absence of nucleotides, there was a weaker binding of the clinal exo to the DNA. And then in the presence of nucleotides, which are in lanes 3, 4, 7, and 8, which we'll see here, there are darker bands, and this shows enhanced binding. Also, the stimulated formation of complexes of different mobility, and those can be found in lanes 7 through 10, right here. And this you'll see in the incubation with PAT, there was some presence of the nucleotides. However, in the presence of the natural nucleotide, there was no bands present. Then with tertiary complexes formed by HIV-1 reverse transcriptase, this was seen that indicates conformational change, lanes 3 through 10. So in lane 1, you're going to have your standard right here. So there's going to be a band because that's just your nucleotide. However, lanes 3 through 10 is the nucleotide triphosphides and then your analog bases. So that does show that there is a conformational change. There was some similarity mobility of PAT containing complex to DTTP containing complex in lanes 3, 4, 7, and 8. So what you'll notice is that when PAT, their bands are darker as the DTTP indicates up here. So in conclusion, the 5-phosphate region of prepared conjugates were crucial to recognize substrates by all DNA polymerases. Upon binding of the PAT, ternary complex of clinal exo polymerase and HIV-1 reverse transcriptase adopt alternative conformations similar to that formed from binding of an incorporation deficient nucleotide. So for future research, discriminative incorporation of PA5T, which is an analog, by HIV-1 reverse transcriptase suggests this compound may be used for antiviral therapy. Exact mechanism of the PAA-induced alterations and the structure of the productive complex is yet to be explained. Purposeful conjugation of these compounds with nucleotides may provide a basis for further development of novel antiviral drugs. These are my references. Thank you for your time.